250 liters of filtrate a day that are being filtered out, which is a staggering number, really, for these really small organs that we have in our body. But of course, what we have to do is we have to reclaim most of that stuff that gets filtered out because we don't want to become dehydrated, right? So we start with that process of reabsorption here in the proximal convoluted tubule. And we said that a lot of that is tied to sodium, right? I think one of the themes that's going to come out of this is that water is always going to follow salt. And we definitely see that when we start to take a look at anywhere, but the kidneys becomes especially. So that's kind of where we left things this morning. We kind of ended at the end of our discussion on reabsorption. So what I wanted to do today, this afternoon, is I wanted to finish talking about this process of urine formation, and also talk about how we kind of alter the composition of our urine based on a couple of particular needs. If you talk to anybody in the morning lab, they'll tell you that I fried their brain a little bit, but that's just propaganda. It's not true. We had a good discussion. So hopefully we can have a good discussion this afternoon as well. But we do need to finish this material. After we've done that, we will talk about things that can go wrong with our bodies and how those things that go wrong show up in the urine. Okay, so what we'll do to kind of illustrate that is we'll do a urinalysis after we're kind of done talking about this. Okay. So before we start, are there any questions from lecture this morning that we need to address? Hopefully it's pretty clear, okay, or seem clear at the moment. I know that we still have to process and study and stuff like that, but hopefully what we talked about felt clear this morning. So what I want to do is I want to continue walking us through the nephron, right? We've gone through filtration. We said that the majority of reabsorption happens in the proximal convoluted tubule. So what I want to do is keep walking us through the nephron. And the next thing that we need to look at is the loop of Henle. Okay? And the loop of Henle has a really specific job, but it's also not immediately obvious, right? Like we don't look at it and say, oh yeah, that makes total sense, right? So I mean, we're going to go over what it does, but we're also going to like have to come back to it in a few minutes and say, oh, that's why it does. All right. Actually, I think I have my diagram up there as well. This diagram just wanted to. So let's go ahead and draw on the big board. Let's go ahead and draw a big loop of Henle, right? So I've got my proximal convoluted tubule right here, and then we have that descending limb of the loop of Henle. We have the hairpin turn, and we have the ascending limb of the loop of Henle. And then we have distal convoluted tubule, and we're going to empty it into the collecting duct right here. So this is kind of the remainder of our nephron. I'm leaving the renal corpuscle off, but we've already gone through that. Okay? So that's part of the anatomy. Okay, when we look at that loop of Henle, regardless of whether it's a cortical or a juxtamedullary nephron, we still have this structure. The other thing too, and the reason I'm showing you guys this, is because I wanted to just kind of briefly remind you that when that efferent arteriole leaves the glomerulus, what it does is it forms this structure. These are called paratubular capillaries. And specifically, those capillaries that are surrounding the nephron are called the vasa recta, right? So we have the vasa recta. If we zoom in on our model here, they kind of show it in a little bit of a funny way, but you can see these capillaries right here. Those are representing the vasa recta. The name vasa recta literally means erect vessel, right? So they're kind of standing straight up. I'm not going to try to draw it, you know, exactly like our model of these diagrams show it. What I am going to do, though, is I am going to include a blood vessel very close to my loop of Henle that will represent my vasa recta. Okay? And it basically is paralleling the loop of Henle. So it's basically doing this. Right? So I've got my loop of Henle here in black, and then because I stole a red pin from the lecture room this morning, I have a red pin back in here. So I'm going to use it today. We've got our vasa recta right there. So we're going to talk about the fluid flow in a minute, but obviously the filtrate, the precursor of our urine, is leaving the proximal tubule. It goes down the descending limb of the loop of Henle, and then it goes up the ascending limb. Okay. The blood in the vasa recta is flowing in the opposite direction. Okay. So the blood in this capillary is actually flowing in this direction. Okay. It's flowing down like this. It goes down through here, and then it comes up through here. So the blood that we find in the capillary very close is running counter current to the fluid in the loop of Henle. We call that counter current flow. It's called counter current flow. And that will become important in the next couple of minutes. And I'll explain why. So that's just a little bit of anatomy. The other thing that we need to do is we need to start to talk a little bit about putting a number on how concentrated this fluid is, right? I think that we can look at our urine and we can say, oh, that's really concentrated urine, or oh, that's really dilute urine. But we need to actually have a number to be able to give that. And physiologists measure concentration, and chemists measure concentration in a number of ways. But really, what we're interested in as physiologists is how much stuff is there in this fluid. Okay? And the units that we give to that is sort of a form of molarity. It's called osmolarity. And the units that we use to describe that are called osmoles. And all of our body fluids are in the thousandths of osmoles, so we use milliosmoles to be able to describe that. So the unit that we use is milliosmoles. Okay? It really is just a measure of the amount of stuff that's dissolved in a particular solution, right? I don't want you to get too hung up in understanding the units, but the easiest way to sort of think about it is to think that if a particular fluid is really, really dilute, like there's not a lot of stuff in it, that number is going to be really low. If there's a lot of stuff dissolved in it, the number is going to be really high. Okay, and that's really all I'm really concerned about, right? Is the low number versus the high. But we don't need to get into the derivation of it or anything like that. We can save that for another day. But if this number is low, we're talking about a fluid that's really dilute. If this number is high, it's a really concentrated solution. There's a lot of stuff. Most of your body fluids, okay, so we're talking intracellular fluid, extracellular fluid, blood plasma, most of your body fluids hover somewhere around 290 to 300 milliosmoles. Okay? So most of our body fluids are somewhere about 300 milliosmoles. 290 to 300 is pretty much the normal range. Okay? Doesn't really affect this class a whole lot, but your fun fact for the day is that tears are like the one notable body fluid that we produce that's way less than that. Tears are what we call hypoosmotic. They're around 100 milliosmoles. No idea why, no explanation for it, but that's it. Right? So, like I said, it doesn't really relate to the lecture today, but maybe you'll be able to answer a Trivial Pursuit question on it later or something. Okay. We all good so far? Any questions? So, kind of continuing with this idea, right? 300 milliosmol, that filtrate that we're producing here is about 300 milliosmoles. Okay? So, at this point in the process of urine formation, the fluid that we're producing is around 300 milliosmol, just like just about every one of our body fluids. Okay? But then, it's going to turn the corner and it's going to start to go down. This is where things get interesting. Right? A couple of quick rules about the loop of Henle. Okay? The descending limb of the loop of Henle is only permeable to water. Okay? So what that means is that the only thing that can move across the descending limb of the loop of Henle is water. Right? It's just the way that the cells are put together, the way that the cells are made. Okay? For the ascending limb, the only thing that can move across the ascending limb are ions. Okay? They're not permeable to anything else except for ions. So we can move sodium, for example, here across the membrane fairly easily. Okay? We can move sodium across. Just like what we saw earlier this morning with sodium in the proximal convoluted tubule, this is active transport. This requires ATP. Okay? So ATP is being used, and we can actively move sodium across the membrane. That's against its concentration gradient. That's why we have the input of energy that allows us to do that. Okay? All right. So let's talk about how this process all comes together. Right? We've got the building blocks all in front of us here. So let's talk about how this works. And let's start with the ascending limb. Okay? I know that I'm skipping this. I'll come back to this. But let's start with the ascending limb. Okay? The ascending limb of the loop of Henle is actively pumping sodium out of the filter. Okay? So if I'm removing stuff from the fluid, is it going to become less concentrated or more concentrated? Become less concentrated, right? So it's going to become less concentrated. And if I'm moving stuff into the blood and into interstitial space, that means the osmolarity of these are going to go up, right? So let's look at the blood. Let's look at the blood right here, right? That blood entering the vasa recta, probably starting out around 300 milliosmoles, right? Just like every other body fluid. Okay. But as that blood travels down, and I start to add sodium to it, what's going to happen to the osmolarity of that blood? It's going to go up, right? At the very bottom, down here, the osmolarity in that blood is quite high. It's around 1,200 milliosmoles. So we've added a lot of stuff to it, right? Okay. And then that blood turns the corner and it starts to go up in this direction. Okay? In the descending limb, my filtrate is coming in at 300 milliosmol. Okay? Is there a concentration gradient between my blood and this space in here? There sure is, right? So what's going to happen? That's going to pull water out of my descending limb. So as this filtrate travels down, it becomes more and more and more concentrated. Does that make sense? Okay. And then you guys just told me what happens, but then on its way up, what happens to the filtrate? It's losing stuff because I'm pumping sodium out, so it becomes less concentrated. So by the time we get up here, our filtrate is only about 100 milliosmoles. Okay. So because the blood and the filtrate are running at opposite directions to each other, it allows us to load the blood up with salt on this side. Okay? Load the blood up with salt, make it more and more concentrated, and then on the way up, all that salt allows me to pull a ton of water out of my descending limb. Does that make sense? Okay. And so by the time the blood gets back up here to the end of the vasa recta, guess what? It's back nice and happy at around 300 milliosmol because I've diluted it. It enters the general nothing has ever happened to that blood. Okay? Okay. Why am I going through the process? Why am I sending this isoosmotic filtrate into the descending limb of my liquid Henle? Concentrating, all the way down to the bottom, this super hyper concentrated filtrate. And then I dilute it on my way up. And actually, I end up with a hypoosmotic filtrate at the other end. Why don't I go with that process? Okay. What I'm doing is I'm doing all of that so that I can pull salt out of my filter, pull ions out of my filter. And not only do I put some of those ions in my blood, I also accumulate those ions in the interstitial space of the kidney. Okay? So I'm accumulating salt in the interstitial space. Anybody eat kidney? No? I have a friend who likes kidneys. But if you ever have the opportunity to eat kidney or taste kidney, one of the things you'll notice about it is it's a really salty. Okay? Very, very salty. And that's because the loop of Henle exists. And we're pulling salt out, right? So up here, where the gradient is not that big, it's a little salty. But the further we go down into the kidney, and we follow that loop of Henle, the saltier and saltier and saltier that interstitial space becomes. Right? That interstitial space becomes super salty the further we go down. The longer your loop of Henle is, the saltier the space becomes. So this is why in animals that have more juxtamedullary nephrons, those kidneys, that interstitial space of the kidney gets really, really, really salty. Okay? Does that make sense? Kind of, sort of? So we're pulling, the whole point of the loop of Henle is to pull salt out of our filtrate and just load the interstitial space up with salt. Okay? A little bit of it goes into the blood, a lot of the water goes into the blood, but a lot of that salt accumulates in the interstitial space, the space around the renal tubule. Okay? This is why when we show pictures of this, and this is a pretty good example right here, you'll notice that what they do when they color these is they use like a lighter color up here at the top, and the deeper you go, it gets darker and darker and darker. Because they're trying to show you that the interstitial fluid, the extracellular fluid, is getting more and more concentrated the deeper we go. Okay? And again, the longer the loop of Henle, the bigger this concentration gradient gets. Okay? For little short loops of Henle, it's not a very big concentration gradient. For little long loops of Henle, it's a really pretty big. Again, probably doesn't seem like a whole lot right now, but it's going to be important in just a couple of minutes, right? But this is what we're doing. Okay? So it seems backwards. It seems like it's not terribly important, but it actually is going to be really important in just a couple of minutes. The other thing that I want to point out is that we haven't done anything to our urine yet, right? But you know from your own personal experience that when you're really hydrated, you produce a lot of really dilute urine. Okay? You know that when you're dehydrated, you produce a small amount of concentrated. Okay? But look at what we've got right here. Regardless of whether or not you've been sitting on the couch drinking water all day, or whether you've been out hiking in the desert for 3 days, regardless of your hydration state, the fluid at this point, which is almost urine, we're almost to our collecting duct, the fluid at this point is all really dilute urine. Okay? And if we did nothing else to it, what we would do is we would urinate a ton of really dilute urine. Okay? So regardless of your hydration state, you start off with a lot of really, really dilute urine. So the loop of Henle feels weird, it feels strange, it's kind of goofy that we're taking this and we're super hyper concentrating it, super diluting it. But the whole point is to be able to load that interstitial or extracellular space with salt. Okay. Yeah.
So it's only diluted because we lost the salt? Or like the salt went out?
So what you're doing is you're actively pumping the salt out. Okay? When you actively pump the salt out, it accumulates in the interstitial space, but you put just enough in the blood that what you're able to do is pull And that's actually reabsorption. We need to reabsorb more water, so that's just more reabsorption.
So why wouldn't we need to bring more sodium back in? So what I'm thinking is like when you work out, you lose salt, so then you need to replenish like electrolytes.
Yeah, we can reclaim a little bit, like, but it's not a lot. You have to have the salt in the interstitial space. Otherwise, the rest of the process is important. I know I haven't told that part of the story yet, but how are you going to replace those electrolytes? You can't save them when you work out, so what do you have to do?
Food, salt.
Yeah, electrolyte drinks,
Yeah.
food, salt, stuff like that, right? You're going to have to bring it in from other sources. Once it's sweat out, unfortunately, we just can't reclaim it. And having this salt accumulate in the extracellular space of your kidneys is like no problem. So it's like half that. I'll explain that. Anything else? It's a good question. All right. Okay. So, I'm not going to spend more than just a second on it, but let's just really mention that third and final step of urine formation, which is secretion, right? We mentioned this morning there are 3 steps to urine formation. We spent probably an annoying amount of time talking about filtration. We talked a little bit about reabsorption. Real quick, secretion, right? Secretion is the opposite of reabsorption. What you're doing is you are moving things from the blood, okay, into primarily the distal convoluted tubule, right? So secretion is kind of happening up here, right? Secretion is happening up here, and you're moving things into your distal convoluted tubule, which is primarily when it happens. Things that are secreted are going to be things like drug byproducts, right? When we take anything, any type of medication, what happens is we have to break it down. Usually those things are too big to get filtered out, so we have to put them in later. So secretion will help with that. We'll talk a little bit more about pH balance and stuff like that, but we can actually use secretion to change the pH of our blood, things like that. Okay. Beyond that, I don't really have a lot to say about secretion, but it just happens primarily in the distal convoluted. Okay. But what I do want to talk about is I want to talk about all of this really dilute urine that we're now passing into our collector. Okay. So what's going on here? Because every time is not a good time for you to pee a lot of really good. Okay. In fact, most times probably aren't the best. Okay, so I don't know why I drew this one here. I thought I might do it. So let's think a little bit about dehydration. Now that we understand osmolarity, right, we can look at the osmolarity of our plasma. Put 290 is better than most osmolarity. 290, 800. So somewhere in that area. Put a few numbers on there. Okay. So dehydration, right? The word literally means losing water, right? Sweat is a great way to become dehydrated. What are some other ways that we lose water? Somebody this morning said cry, and I was like, every day? Some days it might feel like that. But like on a daily basis, just normal everyday basis, how might you lose water? Obviously, urination is one. Let's take that out of the equation. How else do you lose water? Sweat is one. That's a big one. What else?
Isolation.
Do I?
Isolation.
Exhalation, yeah, exhalation is a big one, right? Every time you exhale, there's water vapor in that breath, right? All you have to do is go camping to realize that. Lock yourself up in the tent for the night. Wake up the next morning and the inside of the tent is all this water condensed on the inside of it, right? So exhalation is a big one, right? The other one that I think sometimes we don't think about is that we actually lose a lot. Joking about crying, but your eyes, right? The surface of your eyes are always wet, right? And there's water evaporating off of them all the time. All you have to do is go outside on a cold morning and put on a pair of glasses or sunglasses, and you realize how quickly they fog up, right? Because all that water evaporating off of your eyes condenses on the lens of your glasses, right? We don't really get cold enough mornings here in Southern California, but occasionally, right? But you do. You lose water evaporating off of your eyeballs. You exhale a significant amount. And you have water evaporating off of your skin. Even when you're not sweating, you still have water evaporation off of your skin as well. Again, go camping. Put yourself in a tight sleeping bag for the night. And you'll realize just how quickly, how much water you can. If you've ever gone camping and woken up to a damp inside of your sleeping bag, that's because of all of the water that you lost. That's a considerable amount of water. Just exhaling alone, you lose about 1 liter of water a day just through exhaling. Okay, so if I wanted to refill this, I could just exhale into it all day long, and I suppose maybe by the end of the day fill it up. It doesn't really work like that. But that's the theory. But you lose about 1 liter of water a day just through your exhaled breath, which is considerable, right? So if you do nothing else except exist, you're eventually going to become dehydrated, right? Even if you're not wasting a lot of water through urination, you will eventually become dehydrated. You lose water. As you become dehydrated, what we see is that you're losing water, but the amount of stuff in your blood remains relatively constant. So what happens to its osmolarity? Your plasma osmolarity will decrease, it will become more concentrated. Okay. You have sensors all over your body for plasma osmolarity, but one of the most important is in a part of your brain called your hypothalamus. Okay? You have a sensor in your brain for plasma osmolarity. It's in your hypothalamus. And so what happens is when your plasma osmolarity increases to such a point where we need to do something about it, our hypothalamus does something about it. In fact, it releases a hormone into our blood called vasopressin or antidiuretic hormone. Which gland secretes vasopressin or antidiuretic hormone? Posterior pituitary releases vasopressin. So what we can do is we can look at your plasma vasopressin levels. Look at your plasma vasopressin levels as you become more and more and more dehydrated. And we see that as the osmolarity of your plasma increases, so do your vasopressin levels. Okay? So if you've been out wandering the desert for 3 days, right? Your best Florence of Arabia impersonation. I come in and take a blood sample. What I'm going to find, almost certainly, is I'm going to find elevated vasopressin levels in the blood. After a workout, after you've exercised, you're dehydrated. I'm going to find elevated vasopressin levels in your blood. Okay. Heck, even just after going to school all day and you haven't drank enough water, I'm probably going to find slightly elevated vasopressin levels in your blood. Okay. Remember, the name of this hormone is vasopressin. It's also antidiuretic hormone. So we said earlier in the semester, a diuretic makes you urinate, and antidiuretic prevents you from doing that, right? So what we're doing here is we're going to be conserving water. So where does antidiuretic hormone go, and what does it do? Okay. Antidiuretic hormone has receptors roughly on the last little bit of the distal convoluted tubule, but also all the way down the collecting. Right? So we can go and look like this. You can go all through within that red box is where we're going to find receptors for antidiuretic hormone. Okay. So that's where it goes. Now what does it do? Okay. Well, what we need to do is we need to take this really dilute 100 milliosmol urine and we need to pull water out of it. By pulling water out of it, what we're going to do is we're going to concentrate the urine. Okay? So this fluid is passing in here, starts off very, very dilute, about 100 milliosmol. Okay? But then if vasopressin is present, we're going to have to use it to concentrate this. How are we going to do that? Gotcha, my board. Not ready to erase this yet. So I'm just going to draw, this is my collecting duct right here. It's a section of collecting duct. All along the collecting duct, we've got the vasopressin receptors. So we've got all of these vasopressin receptors kind of all along there, right? These are all vasopressin receptors. All along. Okay? As vasopressin is released, okay, as vasopressin levels increase and as they go up and up and up, what they're going to do is they're going to come along in the blood supply and they're going to start to bind to receptors. Okay? So vasopressin receptor, so vasopressin is coming in and it's binding to all of these receptors. And when vasopressin binds to its receptors, what it does is it affects proteins called aquaporin proteins. It affects proteins called aquaporins. Aquaporin, that word literally means water core. Okay? Remember, cell membranes have a fatty core. Water can't cross cell membranes very easily. It's got to have a way across. And more than often than not, we have this whole family of proteins called aquaporins. Okay? Aquaporins are water pores or water channels. And up to this point, we've talked about ion channels a lot, right? And we've talked about like sodium channels, for instance, right? They open or they close, right? They open or they close. Aquaporins are a little bit different. They're always open. So if we don't want to use them, what we have to do is we physically have to take them off of the membrane, right? So what we can do is I've got aquaporins kind of floating around in my cell membrane, in my plasma membrane. And if I want to use it, I put it on the membrane, and the water can flow across the membrane. If I don't want to use it, I have to remove it, and then water can't flow across the membrane. And if you use it, put it on the membrane. It makes that sound. You know that? That's a fun fact. It makes that sound when we put it on the membrane. Right? They come on, they come off, right? So what happens is that when vasopressin starts to bind to these receptors, what we do is we're putting aquaporins on the membrane. Okay? So when vasopressin comes in and binds to this receptor, what we do is we put an aquaporin on the membrane. When vasopressin comes in and binds to the receptor, we put an aquaporin on the membrane. When vasopressin comes in and binds to the receptor, we put an aquaporin on the membrane. Okay? Now water can flow freely across the membrane. So let's come back. You guys tell me, do we have a concentration gradient that allows us to do that? So now, what I've done, vasopressin has been released, and now I've come on my membrane, and I've put water pores on it, put aquaporins. Right? I put my water pores on my membrane. super concentrated here, right? Lots of water, little solutes, right? Out here, remember what I did? I made this salty because of my loop of Henle. So is there a concentration gradient? There sure is. Now, guess what water can do? Water can go whoop, whoop. It makes that sound too. Right out, right? So what I'm doing is I'm pulling water out of my collecting duct, right? So now my urine becomes more concentrated. Okay. The more vasopressin there is, the more water I pull out. Okay. So when I'm really dehydrated and there's a lot of vasopressin, this whole thing is filled with aquaporins, and I can pull out water all the way down. So the maximum I can get my urine up to is whatever the salt concentration out here was, which in the case of humans is around 1,200 milliosmoles. So like your stinkiest, most concentrated, most dehydrated pee you ever made is around 1,200 milliosmoles. Okay. The most dilute urine, like the water that you're going, when you're sitting on the couch drinking water all day and you're running to the bathroom every 15 minutes, it's around 100. Pretty hypoosmotic. Okay. So you can vary quite a bit. Do you see why we have the loop of Henle now? You've got to have the salt out here to be able to pull the water out of the collector. Okay. If the salt isn't here, then there's no water coming out of the collector. And you're not going to be able to do anything with your urine because you're going to be dehydrated very okay. The longer those loops of Henle are, the more water you're able to pull out, which is why kangaroo rats are so good at concentrating their urine, right? All these really long juxtamedullary nephrons, they're pulling a ton of water out of their urine, okay? And their vasopressin levels have got to be through the roof, too. But their vasopressin levels have got to be sky high. Just now thought of it. Does that make sense? Okay. But if you don't have vasopressin, guess what? That membrane is basically impermeable to water, and no water's coming out of your but only because the loop of Henle set the stage with that salt. Fill in your notes. Just a second.
What enters this? When you drew the arrows from
On the other side of the board.
where it says 100 milliliters.
So we opened the aquaporins, so we actually have water flowing.
Okay, just water.
Of a cell membrane. The beauty of a cell membrane is that they're impermeable to water, right? So if we don't unlock the cell membrane by adding those aquaporins, we just lose a ton of really good urine. Any questions about that? So one of the things that we do is we do sense plasma osmolarity. Like I said, as you become more and more and more dehydrated, your vasopressin levels go up. Questions about that? Yeah. The absence of vasopressin. I honestly don't know what the mechanism is. I've always, I guess, just assumed that when vasopressin goes away, the aquaporins just fall off. But I don't know. There's got to be more to it. I just don't know what it is. I'm basically taking water. That seems comical almost, right? Like vasopressin goes away and it just falls off. I guess that's what happened. Okay. Questions? I'm going to save this in case I'm going to but I'm going to turn the board back around. We need this appear. So one of the things that our kidneys do is they do work with water balance, and I think one of the most obvious things is basic dehydration. Right? But we listed a few other things that our kidneys do too. And one of the things that they do is they help to regulate blood pressure. Okay, so I want to talk about blood pressure right now. And I think at this point in the class, we all can agree that you've got to maintain blood pressure, right? If blood pressure drops too low, like, it's curtains for you, right? You got to keep blood going to your brain, otherwise things are not going to end up very well. So how do we deal with low blood pressure? Okay? Let's talk about that next, right? We've got a situation where blood pressure is low. Okay? Oh no, what we're going to do, right? Okay. Oh no, blood pressure is low. Blood pressure can be low from a lot of reasons. One of them is just dehydration, okay? So it kind of goes hand in hand to some degree with what we just talked about, right? Remember, you have three fluid spaces in your body. You have your blood plasma, you have your extracellular fluid, you have your intracellular fluid, right? So if you're dehydrated and you're losing water, that water has to come out of one of those spaces. And that means that blood volume, blood plasma volume is going to decrease. If that happens, blood pressure also goes down, right? So often with dehydration, we see a reduction in blood pressure because, you know, water has to come from somewhere. There's no magic here, right? So the water has to come from somewhere. Obviously, it can come from other things as well, but one of the big things that it comes from is dehydration. Okay. But again, we have to remedy this. Okay. So how are we going to do this? If you're thinking, can't we just use vasopressin? Yes. Okay. But there's a lot more to it. Right? In fact, this is probably one of the most powerful physiological responses that our body has. It's what happens when our blood pressure drops. Okay. So, how do our kidneys know of blood pressure? Lucky for us, there's blood vessels here. How's that look? How do our kidneys know? Sorry, it's like a hot mess. There we go. Okay. How do our kidneys know our blood pressure is decreased? Turns out I've got an arteriole right here. It's going to tell me that. Okay? It's in my granular cells. Remember those granular cells that we find? Those granular cells that we find right in this region of our afferent arteriole. Those granular cells are going to tell me blood pressure. Okay? So our kidneys know blood pressure is decreased because the granular cells sense it. Okay? There's a few other sensors in our body. The granular cells are kind of the biggie in this scenario. So the granular cells sense a decrease in blood pressure. So what are they going to do? Okay? They are going to cause the kidney to release a protein called renin into the bloodstream. So I stole my red pen from lecture this morning. I'm going to use it. So there's my blood, right? So the kidney is going to release a substance called renin into the bloodstream. Decrease blood pressure, and we see that the kidney, right here, is going to release renin into the bloodstream. What does renin do? Okay. Renin is a protein that is going to activate something that's floating around in your bloodstream right now. You have a protein in your blood called angiotensin 1. Okay. Did I say something about it? Do you know where I'm going with this? Oh, okay. It's called angiotensin 1. Okay? So right now, you have a protein floating around in your blood called angiotensin 1. Could be, under normal circumstances, it is completely inactivated, it does nothing. Okay? But when renin is released into the blood, it becomes active. Okay? So renin activates angiotensin 1. I guarantee many of you guys have heard of this pathway. Let's see why it was told. Usually we abbreviate angiotensin as AMG, and then we use that Roman numeral. Okay. When angiotensin 1 becomes activated, there is another protein, an enzyme in your blood that immediately converts it to angiotensin 2. Okay. So when angiotensin 1 becomes activated, it becomes immediately converted to angiotensin 2, or Ang2. The enzyme responsible for converting angiotensin 1 to angiotensin 2 is an enzyme called angiotensin converting enzyme. The enzyme is called angiotensin converting enzyme. I like enzymes. We managed not to mention too many of them this semester, but this one is an important one. Angiotensin converting enzyme. We abbreviate that ACE, or we call it ACE. Oh, I heard a light bulb go on. You see where I'm going with it? Yeah, right? Okay, so angiotensin converting enzyme converts angiotensin 1 to angiotensin 2. Why is this important? Well, remember, the symptom is low blood pressure, so we need to get our blood pressure back up. Okay, how are we going to do that? Well, angiotensin 2 has an answer for you. Okay, angiotensin 2 is exceptionally good at raising our blood pressure. Angiotensin 2 does a number of things. One of the things that it does is it does stimulate vasopressin release from the posterior pituitary. So we see here that there's more than one type of stimulus, right? One stimulus is an increase in plasma osmolarity. The other type of stimulus is angiotensin 2, right? That will stimulate vasopressin release. As we just discussed, if vasopressin is present, you pull more water from your collecting duct, that increases your blood volume, that increases your blood pressure, okay? But angiotensin doesn't stop there. Angiotensin 2 doesn't stop there. It does a lot more, okay? Another thing that it does is it stimulates your sense of thirst. It tells your brain, I'm thirsty. If you feel thirsty, it's entirely likely that you're going to go get a drink of water. And if you're putting water into your body, it's got to go somewhere. It's going to go to your blood plasma first, and it's going to increase your blood pressure. Okay. You can feel it's really powerful at stimulating your sense of thirst, actually. Okay, so angiotensin 2 stimulates your sense of thirst. Okay. Angiotensin 2 is a really powerful vasoconstrictor. My blood pressure is low. All I have to do to increase it is squeeze that blood with those arteries, and my blood pressure is going to go up, right? So angiotensin 2 is a really powerful vasoconstrictor. Okay. It also acts on the heart as well by increasing cardiac output. Okay, so angiotensin II works on the heart by increasing cardiac output. Which will also increase your blood pressure. Okay? And then finally, the last major thing that angiotensin II does is it causes the release of a second hormone called aldosterone. Does anybody remember which gland secretes aldosterone? We talked about that one earlier in the semester. I promise this isn't a comprehensive class, but it's kind of a comprehensive class because these things come back to haunt us, right? Say adrenal cortex. Adrenal cortex, good job. Yeah, it's the adrenal cortex that secretes aldosterone. It's one of the hormones secreted by the adrenal cortex, right? But in just a second, I'll talk about what aldosterone does. But you can see here that angiotensin II has effects on all of these different organ systems, right? Stimulates vasopressin release. That's going to cause us to absorb more water. It's going to cause an increase in blood pressure. Aldosterone, I'll explain it in more detail in just a minute, but increased water absorption, increased blood pressure. Thirst, you're putting more water into your system, you're going to have an increased blood pressure. Blood vessel constriction, heart beats harder, all of that's going to increase your blood pressure. If you are a patient who has high blood pressure for other reasons, which is not an uncommon thing at all, okay, probably the last thing you want in your body is angiotensin II because it does all of this stuff. So how can we prevent angiotensin II from showing up in your blood? We can inhibit this enzyme, angiotensin. There's an entire class of drugs that I'd be willing to bet that you've heard of called ACE inhibitors. ACE inhibitors are a class of drugs that basically turn this enzyme off. And what they do is they prevent the conversion of angiotensin I to angiotensin II. If we can prevent that chemical reaction from occurring, none of that's going to happen. Okay? Because angiotensin I can't do that. It's only angiotensin II. Pretty cool, huh? Because I bet you guys have heard of ACE inhibitors before. Okay. How we doing? We're almost there, I promise. I only have about 8 or 9 more pages of notes to go. I actually do, but I'm going to shorten them for today because I know our brains are fine. Then we'll take a little bit of a break and then we'll do other stuff. Okay. So real quick, before we move on, let's go ahead and talk about aldosterone. Okay. What does aldosterone do? Okay. It is an adrenal cortex hormone. We did talk about it earlier in the semester, but what does it do? Real quick, back to our back over here real quick. So remember vasopressin had receptors kind of on the distal end of the distal complex. Basically all of the collections, right? Aldosterone receptors are a little bit more localized. They're on the end of the distal convoluted tubule and just kind of the very top of the collection. Right? So this area, let's go ahead and highlight that in blue really quick. That area, kind of right through here, is where our aldosterone receptors are at, right? Like the upper third or so of our collecting duct is kind of where we're at. So this area, kind of right in here, is where our aldosterone receptors are. Okay? So they're not as widespread as vasopressin receptors. They're a little bit more localized. But what does aldosterone do and how does it work? Okay? What aldosterone does is it binds to cells in that area called principal cells, or sometimes we just abbreviate them as P cells, which is funny because we're talking about P. So, no, that's funny, right? Yeah. That's how I remember them anyway. So, on that area, on the membranes of the distal convoluted tubule, and in those you have these cells called principal cells. So, aldosterone binds to cells called principal or P cells. Binds to these cells called principal cells or P cells. And what it does is it actually causes us to actively reabsorb sodium. Okay, so there's sodium reabsorption that happens here. Okay, so it causes us to reabsorb sodium. But do you guys remember what I said about sodium reabsorption? What's going to follow it? Water, right? Water follows salt, right? So when we reabsorb sodium, water follows by osmosis. So it's not a direct reabsorption of water, but it does end with the reabsorption of water. So it's a really powerful in that regard. Just really quickly, I'm not going to test you on this part of it or anything like that, but we are reabsorbing sodium here. And that can have an effect on our nervous system. We know, I think at this point, that sodium plays a huge role in action potentials and stuff like that, right? The way that we reabsorb sodium is through a protein where sodium goes in one direction, but potassium goes in the other direction. So if you have large amounts of aldosterone in your system, you're reabsorbing a lot of sodium, but you're also excreting a lot of sodium that will or potassium that will ultimately end up in your urine. And that can be somewhat problematic because it can throw off the balance of sodium and potassium in your body, and that can lead to things like central nervous system depression and stuff like that. So it's something. If you have a patient who's on high blood pressure medication and you're trying to suppress this, you have to pay a little bit closer attention to sodium and potassium levels in their blood. And I meant to look it up over lunch and I forgot, but I'll see if I can remember today. But again, I'm not going to test you on it, but sodium and potassium both come into play. It's just that we have a lot of sodium in our body, we don't have that much potassium. So if I'm doing a lot of this and I'm actually keying out a lot of potassium, that can cause all sorts of issues. And it's something to think about. Like I said, it's something I don't have straight in my head. I need to think about it a little bit, or I need to look it up. But it is kind of an important thing. So, but for now, this is all we really need to know. Okay. We're almost there, I promise. Okay. Any questions about concentration of urine? How we alter that? Vasopressin is the primary way, but aldosterone can do it too, but that's more tied to blood pressure, okay? Okay, talked about blood pressure, all right? The last thing I want to talk about, and then I promise I'm done, and we'll make this very quick, like painfully quick, is I want to talk about acid-base balance, okay? Because pH of our blood is like really, really important. We covered most of this, I guess on Wednesday, we covered most of this when we talked about CO2 transport in the blood, okay? That's where most of your acid comes from. That's how the respiratory system handles like 80% of your body's acid-base balance, okay? Because most of your acids come from CO2 production. And remember that if you have acid, if you have hydrogen ions building up because of hypoventilation, right, that's called respiratory acidosis, okay? We do occasionally, though, end up with extra acids in our blood from metabolic sources, and if we do, we call that metabolic acidosis. The thing that comes to my mind immediately is lactic acid buildup from heavy exercise, right? When you have that happening, lactic acid levels go up in your blood, that will drop the pH of your blood, okay? Your respiratory system can manage that to a degree, but it has its own problems, right? Your respiratory system is dealing with all the CO2 that you're producing, which also happens to increase when you exercise, right? So we have to do something about changes in acid levels in our blood and a drop in pH that come from metabolic sources. That's where the kidneys come. Okay. Real quick, okay, I'm going to make this as quick as we possibly can. If you are have an abnormally high level of hydrogen ions in your blood. In other words, if you're in metabolic acidosis, your kidneys can actually secrete or excrete, they can excrete hydrogen ions in your urine. In other words, you can basically pee acid. Your urine can become more acidic and the pH of your blood should come back up to more neutral levels. Okay? If the pH of your blood becomes abnormally basic, which is not a terribly common thing. We don't really have naturally occurring basics that occur in our blood. But if that were to happen, you could actually secrete hydroxide ions into your urine, and that would remove them from your blood, and then your blood would become more basic. Okay? I could talk a lot more. We could talk all day about acid-base balance, but I know your brains are fried and we have other stuff to do, so I think that'll be all right. Oh, God, I forgot to talk about something. Oh, well, I won't put it on the test, and I won't even bother you. I was going to talk about how we urinate, but I forgot to even mention it. So we'll just scratch it. But the changes that we see with acid-base balance in the blood and the kidney regulating those are not as fast as the lungs. I mean, the lungs can enact changes to pH within seconds, right? All you have to do is hold your breath for 30 seconds and the pH of your blood changes. All you have to do is hyperventilate for 30 seconds and the pH of your blood changes. It takes like a day or a day and a half for your kidneys to catch up with things. It's not a super fast thing. It can be really powerful in the long run, but it's not a very fast process at all. Okay. All right. Cool. I did it. In an hour and 15 minutes. Not bad. Okay. I need 3 hours of lecture a week. I really do. This needs to be a 5-unit class. I need like on Wednesdays and Fridays. I need an hour and a half of lecture and then 3 hours of lab. I know what you're thinking. I know you would do anything in the world to spend an extra half hour a week with me. But it would be worth it, okay? I can see it in your guys' faces, but it would be worth it. It would be really good. But I need an extra hour of lecture a week so we can get through stuff like this. And if it was only a half hour on any given day, you wouldn't even notice it. You wouldn't even notice it. All right. Any questions? I'm sure there will be.